Good afternoon once again and welcome to your class in NatSci 22. And for this video, we're going to talk about the structure of DNA. Last time we talked about the Mendelian genetics, the study of genetics, and how is plant and animal breeding done. And you will have a laboratory exercise on that on the Punnett square. So for after that, we will go into the molecular detail of genetics, which is knowing the structure of the DNA. And of course, we know what is DNA, or deoxyribonucleic acid, and it is often called the blueprint of life. In simple terms, contains the instruction for making proteins within the cells. So according to the Watson and Crick's model, it doesn't show here, but this is based on the Watson and Crick's model. And we study the DNA for many reasons. Its central importance to all life on Earth, and all living organisms contains DNA, including humans and higher forms of mammals. And also for medical benefits such as cures for diseases and for better food crop production, of course. So let's take a look at our chromosomes and DNA. So our genes are our chromosomes, which are made up of chemical code DNA. So the DNA is a very long polymer. The basic shape is like twisted ladder or zipper, which is called a double helix. The DNA double helix has two strands twisted together, and one strand of DNA is the backbone of the molecule, alternating phosphates and deoxyribonose. The teeth are the nitrogenous bases. So ATP, we know that is the adenosine triphosphate, and the structure looks like this. So one strand of DNA is a polymer of nucleotides, and one strand of DNA has millions of nucleotides. And it has four nitrogenous bases: C for cytosine, T for thymine, A for adenine, and G for guanine. So there are two kinds of bases: the pyrimidines, which are single ring bases, and the purines, which are double ring bases. The thymine and cytosine are pyrimidines. The thymine and cytosine have one ring of carbon and nitrogen atoms, as shown on the figure. And while adenine and guanine are purines, the adenine and guanine each to have two rings of carbon and nitrogen atoms. So we have here shown on the figure. And remember, DNA has two strands that fit together, something like a zipper. The teeth are the nitrogenous bases, but why do they stick together? Because of the hydrogen bonds. So the bases attract each other because of hydrogen bonds. And hydrogen bonds are weak, but there are millions and millions of them in a single molecule of DNA. And the bonds between cytosine and guanine are shown here with the dotted lines. So hydrogen bonds, when making hydrogen bonds, cytosine always pairs up with guanine or CNG, and adenine always pairs up with thymine or adenine and thymine, and is bonded to thymine here. So according to Chargaff's rule, adenine and thymine always join together, and cytosine and guanine always join together, or ATCG. So remember always the bases, which are ATCG. So each cell has about 2 meters of DNA. Can you imagine that? And the average human has 75 trillion cells. Well, the average human has enough DNA to go from Earth to the sun more than 400 times. And DNA has a diameter of only 0.000002 m. So the Earth is 150 billion um, meter or 93 million miles from the sun. So this is what look like the our DNA. So how does DNA replicate? So let's begin with the history. So early scientists thought that protein was the cell hereditary material because it was more complex than DNA, and proteins were composed of 20 different amino acids in long peptide polypeptide chains. So Fred Griffith worked with virulent S and non-virulent R strain in pneumococcus bacteria, and he found out that the R strain could become virulent when it took in DNA from a heat-killed S strain. So the study suggested that DNA was probably the genetic material. So he had this, Griffith has this experiment in mouse, and chromosomes are made up of both DNA and protein. So experiments on the bacteriophage viruses by Hershey and Chase proved that the DNA was the cell's genetic material indeed. So in this, the radioactive 32P was injected into the bacteria. So the discovery of DNA structure actually when Erwin Chargaff showed the amounts of the four bases of the DNA, ATCG. In a body or somatic cells, it has 30.3% adenine, 30.3% thymine, and 19.5% guanine, and 19.9% cytosine. Adenine must pair with thymine and guanine must pair with cytosine according to Chargaff's rule, and the bases form weak hydrogen bonds. So according to Rosalind Franklin, which took diffraction X-ray photographs of DNA crystals in the 1950s, Watson and Crick built the first model of the DNA using Franklin X-rays. So this is the X-ray of Rosalind Franklin, and therefore the DNA structure made up of two strands called a double helix, which with sides made of a pentose sugar deoxyribose bonded to phosphate groups by phosphodiester bands, and the center is made of nitrogen bases bound together by weak hydrogen bonds. So again, this is a DNA double helix, and the rings of or rungs of ladder and the nitrogen with the nitrogen bases ATGC, and the legs of the ladder with the phosphate and sugar backbone. So helix in most DNA has a right hand twist with 10 base pairs in a complete turn, while the left twisted DNA is called the zDNA or southpaw DNA. Hotspots occur where right and left twisted DNA meet, producing mutations. So the for DNA stands for deoxyribonucleic acid and made up of this subunits nucleotides, which are made up of phosphate group, 5-carbon sugar, and nitrogen base. So the DNA nucleotide structure is shown here. So the pentose sugar are carbons which are numbered clockwise from 1 to 5 as shown in the figure here. So this is the DNA structure again, and it has anti-parallel strands. One strand of DNA goes from 5 to 3 sugars, the other strand in the opposite direction going 3 to 5 sugars as shown on the figure here. So the nitrogenous bases, double ring purines, are actually adenine and guanine again, and the single ring pyrimidines, the thymine and the cytosine. But although adenine always pair with thymine and cytosine always pair guanine, but they have the different nitrogenous bases, double rings and single rings. So the base pairings, purines only pair with pyrimidines, and 3 hydrogen bonds required to bond guanine and cytosine as shown on the figure here. And 2 hydrogen bonds are required to bond adenine and thymine as shown on the figure here. So unlike the thymine the adenine and thymine, there are only 2 hydrogen bonds, but for guanine and cytosine, there are 3 hydrogen bonds. So is there a 30 if there is 30%, then adenine, how much cytosine is present? So of course there would be 20% cytosine with 30% adenine, thymine, and 20% guanine, cytosine. Therefore, 60% is adenine, thymine, and 40% uh, cytosine and guanine. So let's talk about DNA replication. DNA normally has to be copied before a cell divides. So DNA is copied during the S or synthesis phase of the interphase. Remember that we have discussed last time on the previous videos on the cell division. So new cells will need identical DNA strands. So in the S phase, during the interphase of the cell cycle, the nucleus of the eukaryotes is where the DNA replication takes place in the S phase. So it begins at origins of replication where two strands open forming replication like in a replication forks or in a Y-shaped region, new strands grow at the forks as shown on the figure here. So at the two DNA strands open at the origin, the replication bubbles form. And in prokaryotes or bacteria, they have a single bubble, while in eukaryotic chromosomes, they have many bubbles. So enzyme helicase unwinds and separates the two DNA strands by breaking the weak hydrogen bonds, and the single strand binding proteins attach and keep the two DNA strands separated and untwisted. And enzyme 2-polymerase attaches to the two forks of the bubble to relieve stress on the DNA molecules as it separates, as shown on the figure here. And before the DNA strands can form, there must be RNA primers present to start the addition of new nucleotides. The primase is the enzyme that synthesizes the RNA primer, and the DNA polymerase can then add the new nucleotide. So this is how the RNA primer, where the origin of replication is, and they are in the center is the DNA helicase. So the DNA polymerase can only add nucleotides to the 3' of the DNA, and this causes the new strand to be built in a 5 to 3 direction, as shown. This is the direction of the DNA polymerase replication. So remember how the carbons are numbered. We have 1, 2, 3, 4, 5. And remember the strands are anti-parallel. And so the synthesis of DNA, the new DNA strands, the leading strand is synthesized as a single strand from the point of origin toward the opening replication fork in this side. So for the synthesis of the new DNA strands, the lagging strand is synthesized discontinuously against overall direction of the replication, and is this strand made up of many short segments replicated from the replication fork toward the origin. So based on the Okazi fragments, this is the series of short-term segments on the lagging strand, which must be joined together by an enzyme. So this is the Ozaki fragment. So the joining of Ozaki fragments is where the enzyme ligase joins the Ozaki fragments together to make one strand. So this is the figure here shows the replication of strands from the point of origin into the replication fork. So what is proofreading new DNA? So DNA polymerase initially makes about 1 in 10,000 base pairing errors. An enzyme proofread and correct these mistakes. So the new error rate for DNA that has been proofread is 1 in 1 billion. So can you imagine that? So 1 in 1 billion base pairing errors. So But there is an enzyme that's gonna proofread and correct these mistakes. So there is what we call the semi-conservative model of replication, which is this idea presented by Watson and Crick. The two strands of parental molecules separate and each acts as a template for a new complementary strand. So the new DNA is now consists of a parental or the original and a new strand of DNA. So, all chemicals and ultraviolet radiation damage the DNA in our body cells. Therefore, cells must continuously repair this damaged DNA. So, excision repair occurs when any of the 58 repair enzymes remove damaged parts of the DNA. With the DNA polymerase and DNA ligase replace and bond the new nucleotides together. So what would be the complementary DNA strand for the following DNA sequence? With a DNA 5 and CGTA TG3, so the answer would be DNA 5 CGTA TG3 and DNA 3 GCATAC5. So let's talk about the DNA technology. So the DNA extraction composed of the chemical treatments cause cells and nuclei to burst. So this DNA is sticky and can be pulled out the mixture. So it's called the DNA spooling. So in one of your laboratory exercise, you can is the DNA extraction, which you're going to do with a in your laboratory exercise, you have there your methods using household and plant source of ingredients. So this is now the photo of your spooled DNA that you would be able to see after your laboratory exercise. So cutting DNA, where restriction enzymes cut DNA at specific sequences are useful to divide DNA into manageable fragments. So this rDNA will be cut into specific sequence by restriction enzymes. So we have the process called electrophoresis, where DNA can be separated based on the size and charge. And the phosphate groups are negatively charged while DNA is placed in a gel and the electricity is run through. So as shown on the photo here, the negative DNA fragment moves towards the positive stage, positive N I mean, and the smaller fragments move farther and faster. So this is the overall procedure for the electrophoresis. So this is the restriction enzymes. Restriction enzymes cleave DNA into smaller segments of various sizes, and the DNA segments are loaded into wells in a porous gel. The gel floats in a buffer solution within a chamber between two electrodes, and when an electric current is passed through the chamber, the DNA segments move toward the positive charge cathode. The smaller DNA segments move faster and farther than the larger DNA segments. So these are the steps in DNA sequencing. So many copies of a single strand of DNA are placed in a test tube. The DNA polymerase is added and a mixture of nucleotides is added, some of which have dye molecules attached. Each base, the ATCG, has a different color dye. And by chance, some dyed nucleotides and some regular ones are added. Then the dye molecules are large and stop the chain from growing. So the result is DNA fragments of multiple sizes with colors that can be identified. After then the gel separates the resulting fragments by size, we read the sequence from bottom to top. And then the DNA is copied through the polymerase chain reaction, also called the PCR, or a method of making many copies of a piece of DNA. So the steps include a DNA molecule placed in a small test tube, and the DNA polymerase that can work at high temperature is added, and the DNA is heated to separate the two strands. The primers were short pieces of DNA complementary to the ends of the molecule to be copied are added. So the tube is cooled and the DNA polymerase adds new bases to the separated strand. So this is what the overall the polymerase chain reaction occur, where there's inside the test tube, the small test tube, it's heated and then the primers are added and then it replicates and replicates and replicates. So large amounts of DNA can be made from a small starting sample into thousands more. So another DNA cloning is clone is a member of a group of genetically identical cells and may be produced by asexual reproduction or mitosis. So as you can see here on the figure is cloning usually occur because in animals, in bacteria, and in plants, we call them it's just very easy through asexual reproduction. So the body cell taken from a sheep is the DNA is extracted and egg cell taken from the sheep, the clivus is removed. DNA from sheep A fused with egg cell and then from sheep B then fused developed into embryo and then placed in the uterus of foster mother and then we have the clone. So cloning organisms requires a body cell from one organism and egg cell from another, which are fused. The resulting cell divides like a normal embryo. So we have the first clone organism or animals named the dolly sheep. So the Human Genome Project started in 1990 and research effort to sequence all of our DNA, 46 chromosomes, which over 3.3 billion nucleotides, and mapping every gene location called loci are conducted by scientists around the world. So the Human Genome Project insights are only 2% of human genome codes for proteins or exons. And other 98% introns are non-coding, only about 20,000 to 25,000 genes expected of 100,000 genes. Proteome, an organism's complete set of proteins, and about 8 million single nucleotide polymorphisms or SNP places where humans differ by a single nucleotide. About one half of genome comes from transposons or pieces of DNA that move to different locations and chromosomes. So the benefits of this project is for the improvement in medical prevention of disease, gene therapies, diagnosis techniques, production of useful protein, products for use in medicine, agriculture, bioremediation, and pharmaceutical industries with improved bioinformatics using computers to help the DNA sequences. So the benefits of genetic engineering, we have here first biotechnology or the use of gene science to create new products from plants and animals, to provide improved food products, medical advancements, and enhance environment. So we have the so-called herbicide resistant crops. These are available in Roundup Ready soybeans, corn, cotton, and canola. So they have the CP for EPSPS or the Roundup Ready gene. So even they are spread with herbicides, so they are very resistant. So the 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 benefit of this is that less you can use less weeding or labor for weeds instead of manual labor. So the biotechnology breakthroughs include insulin in 1982. This is the first commercial biotech product which is reliable, inexpensive, and can be source of insulin for diabetics. And rice enriched with beta-carotene and iron by the International Rice Research Institute, and bananas contain edible hepatitis vaccine, and potatoes with higher solid content, garlic that lowers cholesterol, fruits and vegetables that reduce risk cancer and heart disease. And for environmental benefits, we have reduced pesticide use, lower energy requirements, cleaner water, and less soil erosion. So thank you very much for listening. And if you have a question, you can ask the questions on the posted video or on our group chat. So I will. This would be our last meeting actually. So for the rest of the topics on protein synthesis, evolution, population genetics, and biological classification and ecosystem, you have to do your own self-study and self-reading. And if you have questions anyway, you can always chat me on our Facebook group. So thank you very much, and I hope I will be posting your final exam as well, and which will be due next week. So I will just wait for your submissions. So thank you very much, and hopefully you will be successful in this semester.